Hello dear students. The next chapter in your syllabus for MB343 metabolism is biosynthesis and degradation. In this chapter we are going to see the synthesis of different polysaccharides, different fatty acids and their degradation. Okay. So synthesis means it is the polymerization process. It is the synthesis process, okay, that is known as biosynthesis. And degradation means to break down, okay, to break down the polysaccharides into monosaccharide. So in short, biosynthesis means to produce and degradation means to degrade, to break down. Okay, so that is the theme of your chapter. The first point in this chapter is chemistry, concept of polymerization of macromolecules. So in that, the first point is polysaccharides, where we will study the synthesis of starch, glycogen and peptidoglycan. Each polysaccharide we will see step by step. Okay, so we'll start from the starch. Before going to the starch, let's understand what is polysaccharides. You might have studied in your previous standards about the polysaccharides. We will just revise the term polysaccharides. Okay, so poly means many. Poly का मतलब होता है many. And saccharides, these are the sugar units. Okay, these are the sugar units. Then it may be single sugar unit called as monosaccharide. It may be two sugar units called as disaccharide, and many sugar units it is called as polysaccharides. Okay, so carbohydrates with more than 20 monosaccharide units are called as polysaccharides. अगर 20 से ज़्यादा units होंगे then it is known as polysaccharides. Majority of carbohydrates in nature usually occurs as polysaccharides. तो नेचर में जो हम देखते हैं that are mostly in nature polysaccharides are been found instead of mono, di or etc. Okay, so in nature mostly polysaccharides are been found. Usually polysaccharides have hundreds or thousands of monosaccharide units. मतलब अगर 20 से ज़्यादा यूनिट्स होंगे, ओके, तो हम उससे polysaccharides बोलते हैं. Not only 20 but it may consist of thousands, hundreds of units, okay, called as the polysaccharides. And if we'll see the classification of this polysaccharides, then they are divided into two groups based on the composition. First is homopolysaccharide and second is heteropolysaccharides. Homo means same, similar. Okay, so homopolysaccharide means these polysaccharides are made up of single type of monomer that are known as homopolysaccharides. Opposite is heteropolysaccharides. Hetero means different. Okay, so the polysaccharides which are made up of different units called as heteropolysaccharides. अब ये homopolysaccharides और heteropolysaccharides where they are being found. So these storage polysaccharides are usually homopolysaccharides, जो storage form of food material होता है, जैसे कि starch. Okay, starch is the storage form of food material which is stored in potato, maize, etc. So starch is an example of homopolysaccharide. तो होमोपॉलीसैकेराइड कहाँ फाउंड होते हैं? इन द स्टोर फॉर्म ऑफ फूड मटेरियल, दैट आर द होमोपॉलीसैकेराइड्स. हेटेरोपॉलीसैकेराइड्स, दिस आर द स्ट्रक्चरल पॉलीसैकेराइड्स. ओके, तो स्ट्रक्चरल पॉलीसैकेराइड्स जो होते हैं दे इंक्लूड बोथ होमो एज वेल एज हेटेरोपॉलीसैकेराइड्स. सो दैट आर द टू क्लासेस. फर्स्ट नाउ वी विल सी डिफरेंट एग्जांपल्स. अगर हम बात करेंगे अबाउट दिस polysaccharides, then different examples include starch, glycogen, peptidoglycan. Okay, these are the polysaccharides जिसका synthesis हम study करने वाले हैं. So let's focus first on starch. Starch, this is the structure of starch made up of glucose subunits. Okay, this is the glucose subunit and it is joined by the bond. Okay, glycosidic bond. That bond is known as glycosidic bond. Let's see the introductory part of starch. Starch, it is a polymer of alpha D glucose. In short, starch क्या है? Starch is a major glucose storage compound present in plant cell or present in plants. अगर हम इसे और सिंपल में बोलेंगे तो starch it is made up of glucose units, that is the starch. Okay, and it occurs in two main forms. First is amylose and second is amylopectin. Amylose, it consists of linear chain of glucose monomers linked by alpha 1,4 glycosidic bond. तो amylose क्या है? It is the linear chain of glucose unit. और वो जो glucose unit है, they are bound by the alpha 1,4 glycosidic bond. And amylopectin क्या है? These are the branched chain of glucose unit and these are linked by alpha 1,4 glycosidic bond as well as alpha 1,6 glycosidic bond. Okay, तो starch ये दो चीजों से बना है. First is amylose and second is amylopectin. The difference between these two is amylose is linear chain linked by alpha 1,4 linkage and amylopectin it is the branched chain linked by 1,4 linkage and that two branch chain are joined by alpha 1,6 glycosidic bond. Where this starch is synthesized? Starch is synthesized in chloroplast in the stroma. Okay, and this precursor is activated by ADP glucose. तो जब starch का synthesis होता है, it requires the precursor called as ADP glucose. Okay, so this ADP glucose means what? ADP, energy that is attached to the glucose unit. तो इससे synthesis start होता है and definitely the enzyme is required called as starch synthase. Okay, so starch synthase enzyme and the precursor, the initiative for the synthesis is required called as ADP glucose, adenosine diphosphate and glucose molecule. So starch is synthesized in this part called as the chloroplast. Other organelles we are not going to focus at the other organelles, just we want to see that where starch is synthesized. It is synthesized in chloroplast. Okay, and temporary storage, अगर हम स्टार्च का टेम्पररी स्टोरेज देखेंगे तो टेम्पररी स्टोरेज इज इन द क्लोरोप्लास्ट. ओके, सो टेम्पररी स्टोरेज इज इन द क्लोरोप्लास्ट एंड लॉन्ग टर्म स्टोरेज इज इन द अमाइलोप्लास्ट. Okay, temporary storage chloroplast and long term storage is the amyloplast. And amyloplast it is a plastid. Okay, as we know that this starch is synthesized in the chloroplast and plastids. Plastid, these are the plant cell organelle. So temporary storage chloroplast and permanent storage amyloplast or long term storage is the amyloplast. And we know that in most plant starch is the main storage form. ठीक है, बहुत से plants में starch is the main storage form, but in few plants like sugarcane, sugar beet, sucrose is the primary storage form. बहुत से plants में starch storage form में होता है, लेकिन in some plants like beet, sugar beet or sugar cane, sucrose is the primary storage form. And like glycogen, starch has the high molecular weight. Okay, so example of homopolysaccharide is the starch. This is the structure of amylose. ये है amylose का structure which is a continuous chain and it is a linear chain, unbranched है, कोई branch form नहीं है, and it mostly consists of 4,000 glucose units which are joined by alpha 1,4 glycosidic linkage. Okay, so this is the first position, second position, third, fourth, fifth and sixth. Okay, यहाँ पर भी देखेंगे 1, 4, 3, सॉरी 1 2 3 4. सो यहाँ का 1 एंड यहाँ का 4. सो दैट इज द अल्फा 1,4 ग्लाइकोसिडिक बॉन्ड. दिस इज द स्ट्रक्चर ऑफ अमाइलोज एंड इट मेक्स अबाउट 20% ऑफ द स्टार्च. ठीक है, मैंने आपको कहा कि स्टार्च के दो कंपोनेंट्स हैं, अमाइलोज एंड अमाइलोपेक्टिन. तो अमाइलोज का कंट्रीब्यूशन कितना है? अबाउट 20% ऑफ स्टार्च इज देयर, सॉरी अबाउट 20% ऑफ अमाइलोज इज देयर इन द स्टार्च. अमाइलोपेक्टिन की अगर हम बात करें तो इट इज द ब्रांच्ड चेन linked by these two branches are linked by alpha 1,6 glycosidic bond, and these glucose units are joined together by alpha 1,4 glycosidic bond. It consists of as many as 10,000 glucose units. Okay, 10,000 glucose units hai. So that is about the amylopectin. If we will see the difference between amylose and amylopectin, then amylose it is a straight chain of glucose unit. Amylopectin it is a branched chain of glucose unit. Amylose contribute 20% of the starch. Contribution कितना है? 20%. Okay, and amylopectin का 80% contribution है. Amylose these are less soluble in water, and amylopectin it is more soluble in water. Amylose it is the straight chain of glucose unit, and amylopectin it is the branched chain of glucose unit. Amylose stains blue with iodine, and amylopectin it shows the reddish brown color when stained with iodine. These are soluble in hot water without swelling. It doesn't swell in hot water, and these are soluble in hot water with swelling. Amylose they do not form a gel when hot water is added to it, and this amylopectin they forms a gel when hot water is added to it. So this is the introductory part of starch. Now what about synthesis? So this is the synthesis of starch. This synthesis we will discuss in our next lecture in detail, how the starch is being synthesized. As I told that it begins from the precursor called as ADP glucose. So it begins from this ADP glucose, and this is the starch synthase enzyme. Okay, ये जो है it is the starch synthase enzyme. So it requires mainly two components. First is enzyme, that is starch synthase, and second is precursor called as ADP glucose. Okay, so these are the active site XA and XB न्यूक्लियोफिलिक अटैक होता है. So this ये प्रोसेस हम नेक्स्ट लेक्चर में डिस्कस करेंगे इन डिटेल, हाउ एक्चुअली स्टार्च हैज बीन सिंथेसाइज्ड बाय यूजिंग एंजाइम स्टार्च सिंथेस एंड प्रीकर्सर ADP glucose, and these are the active sites present at the enzyme. Okay, so I hope it might be clear to you. In next lecture we will discuss synthesis part. Thank you.